Hi everyone, myself Ms.T.Swetha, working as an assistant professor, Department of Microbiology, KSR College of Arts and Science, Autonomous. Today we are going to discuss about the topic isolation of rhizobium from root nodules and the mass multiplication and mass production and the field application of rhizobium. Okay, before entering into the topic, we have to know about what is microbiology. Microbiology is a branch of science which deals with the microorganisms such as bacteria, viruses, algae and fungus. Okay. Next, the study of their interaction between microorganisms and biotic and abiotic components in environment. Microbes are small living creatures we can't see without naked eye. Microorganisms are small living creatures we can't see by naked eye. We can only see under the microscopes. Like bacteria, viruses, fungus, protozoas. Biofertilizers are substances containing beneficial living microorganisms which enhance the soil fertility and promote plant growths. They are working by increasing the essential nutrients for the plant directly or indirectly and through nitrogen fixation or by stimulating the plant growth. For example, for some biofertilizers, bacterial biofertilizers, azospirillum, rhizobium, fungal biofertilizers, mycorrhizae, algal biofertilizers, PGA, blue-green algae. Rhizobium is the bacteria found in soil which helps for the fix the atmospheric nitrogen into the plants. It attaches to the roots of the leguminous plants and produce nodules. These nodules fix the atmospheric nitrogen and converted into ammonia. That morphology of rhizobium. They appear in elongated rods in microscopic view, and they are gram-negative and motile organism that can move one place to another place by using their flagellum, and they are aerobic organisms. Aerobic, now we are going to see about how to isolate rhizobium from the root nodules. First we have to uprooted the leguminous plants and then the root was washed with tap water. Then the roots are, first we have to uprooted the leguminous plant system, then the root system was washed with tap water thoroughly. Then we have to collect the root nodules by using forceps. Then the collected nodule was immersed in the sterilizing agent. The sterilizing agents are 0.1% mercury chloride and 3 to 5% of hydrogen peroxide, which is used for remove the soil particles and unwanted microorganisms from the surface of the root nodules. Then the root nodules are washed with 70% ethanol. Then they are washed with distilled water. By using mortar and pestle, we have to crush the root nodules to make it paste. Then the paste are subjected to cereal dilution. After the cereal dilution, we have to prepare suitable media for the rhizobium. The suitable media for rhizobium is EMA medium, yeast extract mannitol agar medium. With this medium, we have to add congorate, which is used for indicator. After the spread plate technique, we have to incubate the plate at 37 degrees Celsius for 24 hours. After the 24 hours, we have to observe the growth of the microorganism on the plate. If the colony is rhizobium, that is a creamy color, white color colonies were produced on the plate. If it is agrobacterium, that is in red color. In this picture, we have to see the creamy color colonies produced on the agar plate. And the below picture is represent the microscopic view of the rhizobium. Now we are going to the mass production of rhizobium. There are 6 major steps involved in the production of rhizobium. The first one is inoculum preparation. The second one is processing of carrier material. And third one is selection of ideal carrier material. And fourth one is preparation of carrier material. Fifth one is mixing the carrier material into the broth culture. And sixth one is preparation of inoculants packet. Okay, first we have to prepare inoculum. Now we are going to inoculum preparation. Prepare the suitable media for the bacterial strain in 250 ml, 500 ml, 3 liter and 5 liters conical flask and sterilized in the autoclave. After the sterilization, we have to inoculate the bacterial culture into the flask. Keep the flask at room temperature in 27 degrees Celsius for 5 to 7 days. After the incubation, we have to observe the results. After the incubation, we have to observe the flask for the bacterial growth using as a starter culture. Now the inoculum is prepared. Then second process, processing of carrier material. A good carrier material is important for a good quality of biofertilizer. Second one is processing of carrier material. A good carrier material is most important for a good quality of biofertilizer. Peat soil, lignite, charcoal and press mud and farmyard manure are used as a carrier material. Selection of ideal carrier materials. The carrier material should be in low cost and they should be locally available, and that have high organic content in with it and that do not contain any chemicals in it. and they have water holding capacity in above 50%. Okay, third, selection of ideal carrier material. They should be cheaper in cost and should be locally available and that have high organic content in it and that do not have any toxic chemicals in it. It should have high organic content in it and that do not have any toxic chemicals in it, and they should have water holding capacity in above 50%. And fourth one, preparation of carrier material. The carrier material are grinded into fine powder. Then preparation of carrier material. The carrier material are should be grinded into fine powder. After that we have to add calcium chloride for the neutralize the pH. The neutralized carrier material then subjected to autoclave. One, mixing of the carrier material into the broth culture. Now the prepared and sterilized autoclaved carrier material are mixed with the broth culture. Sixth one, preparation of inoculant packets. The sterilized and autoclaved carrier materials are already mixed with the bacterial inoculants. They are packed and sealed with aseptic condition. These all are the six major steps involved in the mass production of the rhizobium. Now application of rhizobium. There are three field application in the application of rhizobium. First one is seed treatment, second one is seedling root tip, and third one is direct field application. First one, seed treatment. First take a sterile plastic bag and add 2 kg of carrier material. Take a sterile plastic bag and take 2 kg of seeds, and to the plastic bag we have to add 10% of sugar solution, which is the sticking agent, and we have to inoculate the seeds into the bag. Then the seeds are dried into the sterile condition. After that we have to apply on the field. Second one is seedling root tip. This method is mainly used for transplanted crops. In this method, 2 packets of inoculant mixed with the 10 liters of water. Then the root portion of the plant was dipped into the water for 5 to 10 minutes. After that we have to transplant it in the land. Okay, last one, main field application. In this method we have to use farmyard manure for carrier material. We have to inoculate the bacterial culture into the farmyard manure and we have to broadcast into the main field. I hope this video is very useful for everyone. We have to see in the next video. Thank you.